The deadliest protein known to man is the botulinum toxin, a powerful neurotoxin produced by the bacterium Clostridium botulinum. With the lethal dose of 100 nanograms, this protein has been characterized as both a biological weapon and a miracle drug. Interestingly, botulinum toxin is used in one of the most popular cosmetic procedures in the world, Botox, and is known to cause paralysis of the injected muscle. How can something deemed so deadly interact with our muscles safely enough to become such a widespread procedure? In this video, we'll be discussing the biochemical mechanism behind this substance, as well as some of the fascinating studies that have emerged in recent years. To understand how botulinum toxin is able to remove wrinkles, we first need to understand how facial wrinkles are formed. Wrinkles are caused by muscle contractions, which are caused by the interaction between the neurotransmitter acetylcholine and the receptors on muscle cells. Under normal conditions, the vesicle containing acetylcholine will fuse with the membrane with the help of the snare proteins, which consists of synaptobrevin, SNAP25, and synaxtin, as we can see here. Acetylcholine will then be expelled into the synaptic cleft between the nerve cell and the muscle cell, causing muscle contractions, which in turn cause wrinkles. So how does Botox get rid of these wrinkles? The toxin binds to the receptor, and the entire toxin receptor complex gets taken up into the cell via receptor-mediated endocytosis. Once in the cell, the light and heavy chains separate, and the light chain interacts with the SNAP25 protein, preventing the vesicles containing acetylcholine from fusing with the membrane. In effect, the botulinum toxin prohibits the release of acetylcholine, which prevents muscle contraction, and in turn, smooths wrinkles. So how does the light chain recognize SNAP25? Performing a series of experiments on the light chain SNAP25 complex, Breitenbach and Brunger discovered an array of exocytes on the botulinum toxin light chain that were responsible for binding to SNAP25. Now these exocytes are also known as areas of enzyme substrate binding outside of the enzyme's active site. The portion of the SNAP protein that is involved in the reaction is known as SN2. The first exocyte, known as the alpha exocyte, was identified via its interactions with the hydrophobic region of the amphipathic SN2 helix. When they mutated hydrophobic SN2 residues, including I156E and M167E, the kcat-Km ratios, used to indicate the catalytic efficiency of an enzyme, were substantially decreased compared to the wild-type SN2. This indicated that without the interactions between the alpha exocyte and the hydrophobic regions of the botulinum toxin light chain, the enzyme substrate affinity would be reduced. The second exocyte, known as the beta exocyte, is located at the C-terminal end of the light chain and consists of a methionine residue, known as MET202, that's surrounded by a hydrophobic pocket of two beta sheets.
The research also showed that this MET202 residue is integral in helping the botulinum toxin bind to SNAP25. When the M202Y was mutated, the catalytic efficiency ratio of the botulinum toxin SNAP25 complex once again decreased as shown in the figure here.
Now that we've gone through the general mechanism of botulinum toxin, let's discuss how such a deadly compound has been deemed safe enough for commercial use. Firstly, a typical Botox injection is around 1 nanogram, well below the lethal dose of 100 nanograms. And secondly, the injection targets a confined area that does not contain muscles that are critical to our survival. However, given its potential as a lethal toxin, many researchers have also been investigating its mechanism of action in hopes of developing a defense against potential biological weapons containing botulinum toxin. Other research is also being done on developing further clinical applications, including calming muscle spasms in patients with epilepsy. Therein lies the beauty of botulinum toxin. Although it can be a lethal poison in certain contexts, it also has much potential for developing treatments to many debilitating diseases.